The focus on this presentation are leaves. Leaves are organs comprised of tissues. These organs are attached to stems on vascular plants. This is the site of photosynthesis. Leaves are the organs that perform photosynthesis. We are going to divide leaves up into categories such as leaf structure, types of leaves, leaf arrangement, leaf venation, and internal anatomy. The basic leaf structure includes two parts: the blade, which is the flattened portion of the leaf, which is usually the body, and then the petiole, which is the leaf stalk, which attaches to the branch. Leaves are divided into two parts or two types, including simple leaves and compound leaves. Simple leaves are not divided into smaller leaflets, while compound leaves are divided into these leaflets. They look like smaller leaves attached to a branch, but this is not a branch. The reason for this and how we know it's a leaf is because of the axillary bud. This is where it detaches. This is where it detaches from the branch. Each leaflet in a compound leaf does not have an axillary bud. So in the fall, when a leaf falls off the tree, the whole compound leaf will remove itself from the tree. And this usually happens with a change of temperature, change of weather, and a little bit of cork or bark is formed between the leaf and the branch. This allows the leaf to fall off. And this is created and done by a hormone in the tree's body. Just like hormones in the human body that create changes in different parts of our life, in the same way God designed trees with different hormones to prepare itself for different stages throughout the year. And this would be losing its leaf for the winter. Here are some pictures of simple leaves compared to compound leaves. The next category is leaf venations. There are three main leaf venations that you need to be aware of. Parallel venations, which we already discussed, are monocots. And then the last two consist of dicots. The palmate, or hand-like, netted venation, and then the pinnate, which is the feather-like venation. The leaf arrangements on the stem focus on alternate, opposite, and whorled. Alternate is where the leaf nodes, the axillary nodes, axillary referring to the sides, terminal referring to the top, axillary nodes are going to not be directly across from each other. They are going to be alternating, taking turns, while opposite is directly opposite each other. Whorled describes having two leaves at each node. Now, the internal leaf anatomy is very important. We are going to focus on this on a worksheet. You do need to be aware of these structures. The top layer or waxy covering is the cuticle. We already learned that the cuticle is a waxy covering over liverworts and mosses to prevent water loss. Then below the cuticle, we have the epidermis. The long cell structures are called the palisade cells or palisade layer. The squishy cells inside, circular in shape, are called the spongy cells. They look like little circular sponges. On both the top and bottom, there will be more at the bottom are the guard cells. The guard cells are the lips that open and close, and the space between the guard cells is called the stoma, or also known as the stomata. And then running through the leaf are the veins, the vascular tissues, the tubes, which will consist of xylem and phloem. Xylem carrying water up from the roots, and phloem carrying food, sugar, down from the leaves, which was made during photosynthesis. As I said, the stomata is the opening, the space where oxygen and CO2 will be exchanged. Plants will take in CO2 for photosynthesis, and then they will breathe out oxygen, as well as water will leave through evaporation, called transpiration. And as I said, the guard cells are those lips, those mouths that open and close. This is the equation of photosynthesis that you need to know. You will need to memorize this equation, and we'll talk about why and how This is a balanced equation, balancing the front part, which are the reactants, and the last part, which are the products. 12 units of water will enter the roots, while 6 units of CO2 will enter the stomata of the guard cells. This is then transported to the chloroplasts, which is the site of photosynthesis, using light to break apart the molecules into individual elements and then reshape them and reform them into sugar, which is C6H12O6, oxygen, so we have 6 units of O2, and then we have transpiration, which is releasing 6 H2O, 6 waters. Both the oxygen and the water will leave the guard cells, the stomata of the guard cells.